actually turning your problem set in now. Meghna has to leave before class is over, so you can just put it on the back table here. Thank you.
Hi. I was doing my problem set yesterday and I had it in a different folder.
Okay.
And I left it at home. Can I send pictures of it after class?
Either to me or Meghna.
Okay.
Yeah, that's perfectly fine. All right.
Hi. I totally forgot about the problem set, so I was wondering if you drop.
Of course.
Like, you do drop.
Yes, I
problem
do.
sets, so it's okay.
And I do take late ones.
Oh, you
so
do? Okay.
yeah, and these problem sets are really to help you prepare for exam 2.
Okay.
So you can just turn it in when you're ready.
Okay.
Yeah.
Thank you.
Yeah. Sure.
Hi. I didn't realize this was due on the day of the test.
Yes.
I didn't see a due date anywhere.
No,
but
that's fine.
It's done.
Yes. Thank you so much. Yes.
And then I think maybe I forgot to tell you, or like maybe you just forgot, but on last Friday, no, it's okay.
Oh, yeah.
October 4th, that was my last interview.
Okay.
Yes.
Great.
So, because I saw a 0 for like the top
Oh,
questions,
yes,
and I was
yeah.
like, oh shoot, like I must have forgotten or something.
Yeah. Could you actually send me an email to that
Yes.
effect? Okay, that'd
Yeah,
be great.
definitely.
And then I
But
can
yeah.
update. Yeah.
Yeah, that's the last one. I
Okay.
should be here for now.
Okay. Awesome. Yeah. I hope it went well.
Yes, it did.
Oh, that's good.
I got in. I'm going.
Oh my gosh, congratulations. That's awesome. Good for you, yes. Yeah. So yeah, just send me an email and then I'll go ahead and send it.
Sounds good.
Yeah.
Thank you for your understanding.
Oh sure, congratulations, yeah.
So we didn't lift it off because of the whole like Dropbox thing for Canvas. So
You dropped it in.
there's no Dropbox. So do you want like to come after class and bring it back here?
Yeah.
Both of us
Or, just send it, take a picture of it and send it to Meghna or I. You don't even have to drop it off. Okay, yeah. I do not, no. All right, everyone. Good morning. I am recording this lecture. If you wouldn't mind getting the top hat devices out, we're going to start a couple of top hat questions.
A lot of them want to know if the answers to the muscle worksheet are online.
Don't know, but I will put them up online.
Okay, also,
Yeah.
were there any extras from the ones I handed out Wednesday? Because some people are looking for them.
Yeah. Yes, there are actually.
Okay.
Oops. There you go.
Oh, are there like, people work them out and then come to class?
Oh, yeah, and I have those over there.
Oh, perfect.
I'll put those in the grade. Yeah. All right. So let's go ahead with the first. I've got 2 questions for you all today. And it pertains to our lecture last time. So, tetanus in skeletal muscle refers to A, this is kind of wordy, so I'll give you a little more time. The maximum contractile force generated as a result of activation of all the muscle fibers within the whole muscle using maximum stimulus intensity. B, the maximum force generated from recruitment of additional cross-bridge interactions between actin and myosin. C, the maximum velocity of shortening resulting from expression of myosin isoforms with high cross-bridge cycling rates. Or D, the maximum force generated by increasing the frequency of stimulation at a maximum stimulus intensity. So this is a little bit more difficult. This is the definition of, one of these is the definition of tetanus. For this one, I'll let you talk to your neighbors if you're kind of, on the fence. All right, we're getting up to about 80 students who have answered.
My phone's having trouble connecting.
Sure.
So can I just write it down?
Yeah.
Okay.
Yeah. All right. Does anyone need more time in this room? Raise your hand up high if you need some time in this room. All right, so I will go ahead and close this. And the correct response is actually D. All right, nice job. All right, so just so you know, this A is actually the definition of the intensity force relationship. That was one of the first ones that we talked about. Tetanus is defined as the maximum force generation by increasing the frequency of stimulation. All right, so this one should actually be pretty easy. Hold on just a second. Let's go to this next one here. In the previous question, I didn't say it out loud, which one of these, tetanus is an example of A, intensity force, B, frequency force, C, length tension, or D, load velocity? All right, we're up to about 80 students. Let's see if we can get more than 85 here. All right, we're up to 85. Anybody in this room need a little more time? All right. So I'm going to go ahead and close this one. And the correct answer is B, frequency force. Nice job. All right. For those that are still a little bit confused about this, the intensity force relationship was actually varying the current past or the shock level of a whole muscle. So if you actually gave a whole muscle a tiny shock, just past a little bit of current, you would only recruit a few muscle fibers. As you increase that shock level, you recruit more and more muscle fibers until you figure out what the maximum stimulus intensity is. That's the current past or the shock level where you're recruiting every single muscle fiber within that whole muscle. That's the intensity force relationship. Frequency force was using a maximum stimulus intensity and increasing the firing rate, the time in between the shocks, right? As you increase that, you get summation. As you increase the frequency, you get summation and what's called tetanus. So again, tetanus is an example of frequency force. All right, so with that, I want to make sure that everyone actually has one of these definition sheets. So this is really going to help. I've had a number of students in my office asking me what is the best way to actually study for exam 2. I think my number one answer is don't cram. All right, don't wait until the last 2 or 3 days to try to fit all of the information into your head. That is not a good strategy. I would say what you could do right now, this is a definition sheet. You should know all of these definitions up until about the frequency force relationship. Okay, so it's important. I would take some time maybe this weekend. Terminology is going to be, a big part of exam 2. I would just make sure that you're familiar with all of these different terms up until today's date, okay? Keeping up on the definitions and going through your slides after every lecture is really a good idea. I mean, the lectures are in video form on the Canvas website. I would just make sure that you fully understand each lecture on a day-to-day basis. Okay, so then when you get to studying for exam 2, you're actually making the connections. All right, so just a helpful hint here, a strategy to help you with exam 2. So I did have a question right before class today. When is exam 2? Because I know we postponed exam 1. I think we're doing very well on time. So I'm going to keep exam 2 at October 28th. All right, so we're going to keep that. We're going to have 6 full days to go through the cardiovascular section. All right, one of my favorite sections. It really brings home, it really kind of connects everything we talked about in cell and neurons and skeletal muscle. And we're bringing it all together and applying it to the cardiovascular section. All right, so with that, let us continue on. Here's where we left off. We're talking now today about the length-tension relationship. We've already gone through intensity force. We've talked about frequency force. I want to remind you of that you need to know what's happening at the molecular level. With intensity force, you're recruiting more and more muscle fibers in that whole muscle until you reach a maximum stimulus intensity. For frequency force, I think this is really important. It is not about additional calcium in the intracellular space. When you increase the frequency of firing, or in other words, you're decreasing the time period in between each of those shocks, what you're doing is you're actually stretching out those elastic elements, stretching them out. They get more and more tight with each shock. And then finally, when you reach tetanus, those elastic elements are as tight as they can be. So that allows for a maximum force generation because all of the force can now be translated at the ends of the muscle when those elastic elements are so tight. And you get a maximum force generation. Okay, so today, we talked about this last time. We actually talked about the length-tension relation, the length of the muscle before you give it a shock, before you shock it. And what you'll notice is there is an optimal length. And I told you before that your skeletal muscle, luckily for everyone, skeletal muscle is attached to your bones at optimal length. So when it contracts, you're basically getting the maximum force generation. So we talked about that, and we also talked about what happens in the heart. At rest, your heart is slightly shorter. Let me show you where you are. In the heart, the length is slightly shorter than optimal length. Okay? It's about here on the graph where this little arrow is. When you start to exercise, you force more blood into the ventricles, which stretches the cardiac muscle to optimal length so you can get a more forceful contraction to compensate for that extra blood that's forced into the heart. So a real distinction between skeletal and cardiac muscle, and it does pertain to the length-tension relationship. So again, what we're doing is we're varying the length of the muscle and then giving it a shock. What you'll actually notice is, you can kind of see that here, you generate a graph that looks something like this. If the muscle is too short here, like this, you can't generate very much force. If you stretch it to optimal length and you give it a shock, it's going to give you that maximum force. And then if you stretch the muscle out too far, like this one here on the very right-hand side, then you can't, again, generate that much force. So that's where we left off on Wednesday. Now I want to talk about what is the molecular basis for that. Let's zoom in on what's happening with those contractile elements within the myofibrils. right? That's where all the actin and myosin are. So what's actually happening is, remember this particular diagram. I just want to make sure that everyone is, I'll give you a little orientation. This reddish, these thick fibers right here, the reddish thick fibers, are myosin. These are the myosin heads. The green thin filaments are actin. And what you'll notice at optimal length, which is number 3 here, if I go back to this graph, we're right here on the graph. Here is where you get the maximum number of cross bridges, where myosin can actually attach to the actin filaments. So you get the maximum number of actin and myosin interactions, or cross bridges, that can give you the maximum force. And that is optimal length. If it's too short, like with number 1 and number 2, you can see that actin overlaps a little bit. So it's hard for the myosin to actually bind to actin, and there's no where for that myosin to go. It's already up against the Z discs. So what happens is you can't generate as much force. That's a problem. Especially with number 1, when the muscle is way too short, you can actually see the myosin heads start to bunch up, and they are not able to actually bind to actin at all. So very little force can be generated when the muscle is too short before stimulation. Again, 3 is the optimal length. You can think about it as basically the number of people playing tug of war, right? The more people at the end of the rope, the greater the force is going to be. You're going to have an optimal, basically an optimal force, maybe even snapping the rope in the middle if you have too many people on the line. But as you reduce the number of people, you're going to reduce the number of force that can be generated. So just a way to think about length tension. The more cross-bridge interactions, the greater the force that can be generated. Now, if the length is stretched out way too far, right? You've stretched that muscle out way too far. What you've done is you've actually pulled the myosin heads away from the actin filaments. And now you can see that there's very little myosin-actin interactions, cross-bridge interactions. And again, you can't generate that much force as a result of that. See, the graph really goes down at number 4. All right, so this is just again telling you this is the molecular basis behind why this graph looks the way it does. And remember, there is an optimal length where you maximize the number of cross-bridge interactions. This is a review slide. Short sarcomeres, actin filaments lack room to slide, so little tension can be developed. Optimal length, lots of actin myosin overlap and plenty of room to slide. Long sarcomeres, actin and myosin do not overlap much, so little tension can be developed. Now, I just want to remind you, the green line right here is actually depicting those active or contractile elements, the actin and myosin. But don't forget those elastic elements. These are the passive elements here. The red line is depicting the elastic recoil effect. That's why you get such an increase when you stretch the muscle out. It's like a rubber band. It's going to want to try to recoil back. And so the actual measurement that you make in the laboratory is the sum of the red and blue line, right? The black line is actually the sum of the active and passive tension. So you get this increase in tension once you stretch it out far enough. All right, there are animations online. If you're actually having any trouble playing some of these animations, you can come up and talk to me about it. I usually use Chrome just to help you out. I use Chrome, and then sometimes you have to go to the address bar and make sure that Flash is not blocked. If you just go up there, sometimes there's a little lock. If you press on that lock, and I can show you, well, I can show you now actually. To these animations, what I might try to do is put these animations online on YouTube. Most people can actually access it on YouTube. So if you actually go to Chrome, and you can see there's a little lock right here up in the address bar. If you click that, a lot of times it'll say Flash is blocked. And then you can actually just click toggle from blocked to allow, and that will help you play those animations. Let me know, and again, here's where that lock is. So let me know if you're having any trouble, but that's one way to be able to access it. All right, so going back, let's go to the load-velocity relationship. All right, so one of these is not like the others. I don't know if you guys remember Sesame Street, but the intensity-force relationship, the frequency-force relationship, and the length-tension relationship are all characterizing muscle using isometric contractions. These are all isometric contractions, and it's really telling you what the muscle is capable of. It's characterizing that muscle in a specific way. Load-velocity relationship is the only isotonic relationship. And what you're doing is you're measuring the distance shortened. Okay? And I just want to show you the two different graphs. Again, this is an isometric recording. You can tell that because tension or force is on the y-axis. You've got a little bit of a latent time. This latent time, this latent period, is due to the fact that action potentials have to run down the T-tubules, activating those dihydropyridine receptors. Calcium floods into the intracellular space and then binds to troponin, and then actin and myosin interact. Cross-bridge cycling occurs, and then you can get contraction. So that's what's responsible for this latent period here. In addition, you can see with an isotonic recording, there's a much longer latent period. Not only do you have to have all of those sequence of events have to occur, calcium has to flood into the intracellular space and bind to troponin, so on and so forth, but the beginning of any isotonic recording is an isometric contraction. You have to generate enough force first in order to pick up a weight. And we know this is an isotonic because the y-axis is distance shortened, the shortening distance. And then again, you have contraction and relaxation. All right, so here is what's happening. I always try to kind of talk about it first. I realized there was an animation. So what you're looking at here, and then I'll show you the animation, is you are looking at an apparatus, a muscle that's being hung in an apparatus. It's attached to a load at the bottom, and when you give it a shock, it contracts and picks up the load. This green line is the light load. The orange line is the intermediate load, so you have a heavier weight attached. And then the purple line is the heaviest load, so the weight is now really heavy. You can see contraction and relaxation. So let me show you what that looks like. You see that. Here we go. So, oops. There we go. Let me pick this up. All right, so what you're looking at here, and there's actually, I'll post this on our website. There's actually some nice questions, that quiz, a little Quizlet that you can take a look at for just to help you study. But what you're looking at, again, is a whole muscle fiber, right? A whole muscle fiber. You have a stimulator and an electrode that's passing current to give the muscle a shock. This is analogous to the motor neuron. Okay, so that motor neuron provides that graded potential that initiates action potentials in muscle. All right, and then you have a ruler here that's going to measure the distance shortened. Okay, so let's take a look first. It's actually going to give you some information about an isometric contraction. Gives it a shock, contraction, and relaxation. Again, this is known as a single twitch. That's an isometric contraction. Let me blow this up so you can see it a little bit better. There we go. All right, so let's do the next one, an isotonic contraction. All right, so here you see an intermediate load. What's going to happen is you're going to shock the muscle and it's going to pick up the load and put it back down. So this is what it looks like. Give a shock, picks up the load, you can see the graph, and then it puts it back down. All right, so that's how that graph is being generated. Now I'm going to click it again. Here's a lighter load, picks it up even farther, so you get an increase in distance shortened, and then you put it back down. And then finally, a very heavy load, right? Give it a shock, and it's so heavy it can only pick it up a little bit and puts it back down. Okay, so you can see the graph that's generated. Now, how this is not anything I would expect of you all right now, but if you've taken physics, how would we calculate velocity? What is velocity? I don't expect everyone to know that question. What is it?
Force over mass.
Not quite. Good guess though. What is it? That's right. It is distance over time. So distance is on the y-axis. Time is on the x-axis. So what we're talking about are the slopes of contraction. You can see here that the slope of contraction is great with a light load. With the intermediate load, the slope is somewhat diminished. And then with a heavy load, the slope is really diminished. Okay, so what we're taking is distance over time, which is the slope. All right, so that's what we do. Distance over time, you can see it's taking the slope and then plotting it. Okay? Taking the slope and plotting it, it's diminished. And then taking the slope of the purple line, and then that's really diminished. Now you can do a curve fitting exercise. Curve fitting exercise, and you can generate this graph. This is called the load velocity relationship. The load velocity relationship. All right, so I don't know if you actually caught this. Let's see here. But let's go to this graph. Now you know how this is generated. When you reach zero velocity, what would you call that? When you get to the point where the load doesn't allow the muscle to shorten, what would you call this at zero velocity? Why don't you talk about it in your groups for a couple minutes? See if you can figure out what's behind that shaded area. When the load is so great that it doesn't allow for shortening, what is that? Anybody want to take a guess? Anybody want to take a guess? What would we call that? If the weight, yeah. That's right. It's an isometric contraction. This is a isometric contraction. So when the afterload, if that weight is so great that it doesn't even allow for shortening, then that is a isometric contraction. So what this is telling you, I'm just going to give you a summary of this, is as you vary the load, you can see that the velocity doesn't stay constant, it changes. As you vary that weight, that afterload, the velocity changes. It does not stay constant. Until you get to the point where the load is so, that weight is so great that you can't even get shortening, you reach the point where that's an isometric contraction. Now, if you even increase the weight, you're going to lengthen the muscle. This would be an eccentric isotonic contraction, a lengthening contraction. All right, so the speed of shortening decreases as the load increases. That's the take-home message. And that video, that animation that I just showed you is to mechanics, and I'll put that on your website. All right, so this one is a tough one to really wrap your head around, all right? But it does give me a chance to give you the definition of preload and afterload. Notice that that is on your definition sheet. Preload and afterload. All right, preload and afterload. All right, what you see here is a first-class lever. It's like a teeter-totter, So you have a pivot point, and then this bar right here moves like a teeter-totter. Okay, so the preload is the weight that you would attach to the muscle to set the length. Remember, this is the length tension relationship. You're using a preload to set the length of the muscle prior to stimulation, before you give it a shock. That is what preload is. Once you set the length of the muscle, then you can give the muscle a shock. When it contracts, it's going to pull the teeter-totter down, and it has to lift a weight. That's called the afterload. Now, I'll tell you what the afterload is. By definition, this is the weight or resistance that the muscle has to work against after stimulation. It's the weight or the resistance that the muscle has to work against after stimulation. So what it does is it pulls down, it pulls the teeter-totter down, it lifts up the afterload, and then you can measure velocity. I'm not going to go into too much detail on this graph here. You won't see this on exam too, so don't try to memorize that. But what you'll notice is optimal length when the preload is setting the muscle at optimal length. You can see the load velocity curve looks like this, where the point where you get to the maximum isometric tension is way out here. You have to go to pretty large afterload force in order to get it to the isometric contraction. If you shorten it or lengthen it beyond LO, you can see how that shifts the curve to the left. It diminishes the cross-bridge interactions, and it basically diminishes the velocity at a certain afterload. So, the main point here is that I want you to know the difference between preload and afterload. Okay, so here's why it's important. When we get to cardiac muscle, I want you to think about preload and afterload. You can actually apply this to cardiac, all right? So preload is defined by stretch of the cardiac muscle, right? And it's basically the amount of blood that you force into the ventricles is setting the length, right? The amount of blood that you force into the ventricles sets the length. That's called preload. What is the afterload? It would be the resistance that the heart has to work against. So it's going to be blood pressure. If you have high blood pressure, then the heart has to work that much harder against that resistance. In fact, if you have unchecked high blood pressure, what happens to your heart? It actually hypertrophies like any muscle. Like if you were strength training, your muscle gets bigger and bigger and bigger because it has to work against such a high resistance or afterload, right? That's a problem. If you have not checked your blood pressure and you're 60 years old, if you go into the hospital and they image your heart and it's twice the size of a normal heart, they say, oh, you've had high blood pressure for a very long time. This is actually kind of the first part of heart failure, okay? So it does have some compromises beating action. So unchecked high blood pressure is a big problem. It causes hypertrophy. All right, so just bringing it back to skeletal muscle, remember when you're strength training, you're building the number of myofibrils within each muscle. All right, so making those connections are important too. So that's the load velocity. Does anyone have a question about load velocity? All right, we've been through, this is really about exercise physiology, but it does apply to the heart, as you've already seen. Do know the definitions of preload and afterload. All right, so let's move on. This is the last part of skeletal muscle. This is the last lecture. Then we're going to move on to cardiac. All right. And I think this is a fun lecture too. I think a lot of students have some physiology. All right. So what we're going to be talking about here is the skeletal muscle and how at first it uses ATP, right? The production and use of ATP is important. As you know, myosin uses a lot of ATP during muscle contraction. So I'm sure this is maybe a good memory, but for a lot of people, maybe it's not such a great memory of Biology 1009 or, a foundational biology class. If you remember the glycolytic pathway and the oxidative phosphorylation pathway, I'm going to bring you back to that. Krebs cycle. All right, so what we're talking about here is how ATP is used and produced. All right, the first one I want to talk about is something called creatine phosphate. Creatine phosphate. So, creatine phosphate is a molecule that's within muscle cells, within muscle, and it can be used to buffer any kind of ATP depletion during intense exercise. How many of you all have been to, say, GNC? And you actually see these huge bottles of creatine phosphate? So what you're doing when you're taking that, a lot of people that work out on a consistent basis will take creatine, is the thinking is it is a molecule that will actually help buffer ATP depletion in the cell and help with muscle fatigue. But a lot of journal articles say that really it only helps for elite athletes who are maybe Olympic athletes, that the creatine helps in the first 6 seconds of their workout. And that's pretty much it. All right, so it can make a difference, like I said, with Olympic athletes. Now, I've had people come up and argue with me after class, but creatine also, people swear that it makes their muscles bigger, right? I'm going to argue that creatine is actually acting as an osmolyte. So if creatine enters into the muscle cells, it will draw water in by osmosis, right? It's acting as an osmolyte. So sure, your muscles do get bigger, but after you stop taking it, you probably lose a lot of water weight. Okay, so, maybe I'm not against arguing with people. I really, if you swear by it, I get it, but it might be a waste of your money is what I'm saying. Okay, so creatine phosphate, something to remember. Remember this molecule, there's an enzyme that actually uses the phosphate group to phosphorylate ADP and then increase ATP within the cell. All right, that's creatine. Now, one thing I just want to mention, when we get to the kidney, we're going to be talking about creatinine, which is a derivative of creatine, but a different molecule. All right, creatinine. So creatine is in muscle cells. It helps guard against depletion of ATP quickly within the first 6 seconds of an intense workout. All right, now there's actually 2 other pathways that I want to make sure everyone understands. Let's start with oxidative phosphorylation first. Oxidative phosphorylation actually uses oxygen, right, proteins and amino acids to generate a very large amount of ATP, right? If you want to review oxidative phosphorylation, back in your 1009 textbook, you can do that. But for our purposes, I want you just to know that this is an aerobic pathway. It uses oxygen, and for every glucose molecule, here's glucose right here, it can generate about 34 to 36 ATP molecules. So it is very efficient. This is the aerobic pathway. However, if you're like me and you go out and you try to run a 5K, you'll quickly go from the oxidative phosphorylation pathway to the anaerobic pathway. And this is the glycolytic pathway. Essentially, you can only, for every glucose molecule, you can only make about 2 ATP, so it's a lot less efficient. And the side product is lactic acid. All right, this is the burn that you feel when you're working out, and that happens during the workout. This isn't the same as the soreness you feel 2 days later. The soreness, remember, are those micro tears within the muscle, right? That helps to build muscle. All right, so that's the anaerobic pathway. Now, any questions about these 3 different uses of ATP, production and use of ATP? Pretty good? All right, what you probably already noticed too is that if you pick up a weight, let's just say it's 25, 35 pounds, you can hold it for a while and you can feel pretty good. You can even get to the point where you reach tetanus, right? Maximum stimulation at a maximum force, right? Maximum stimulation with maximum frequency. But after a while, you can see that your muscle starts to give out, right? It can't generate that force after a long period of time. This is what's known as fatigue. So your muscle will fatigue. And if you don't allow a sufficient rest period, you can reach tetanus again, but even quicker, in a shorter time period, you will start to fatigue again if the rest period isn't sufficient enough. So, in skeletal muscle, repetitive stimulation leads to fatigue and reduced tension. Rest overcomes fatigue, but fatigue will reoccur sooner if inadequate recovery time passes. All right, so what is fatigue? You might assume, and that's rightly so, fatigue is when ATP is depleted, right? There's some of that. It is when ATP actually gets to lower levels. But it's really about potassium gradients actually. When if you're exercising intensely, lots of potassium floods out into the intracellular space and into the blood. Your potassium levels actually go pretty high. And think about this, the potassium levels in the T tubules, because that's such a small space, can actually change those gradients pretty profoundly. And that is one of the reasons why fatigue is a problem as well. That contributes to fatigue. It really takes the pump to reestablish the gradient to overcome fatigue as well. There's a nice article already on your website from Clausen that actually details it's a lot more complicated than people think, fatigue. It talks about the pump, the potassium gradient, ATP depletion. There's a lot of factors that contribute to fatigue. All right, so let's talk about now the different muscle fibers in your body, right? Different muscle fibers. You have slow oxidative skeletal muscle. You have fast glycolytic muscle fibers. And then you have something intermediate called fast oxidative, sometimes it's called FOG, fast oxidated glycolytic fibers. All right, so let's go through that pretty systematically. We have about 5 more minutes. Slow oxidative skeletal muscle don't fatigue easily. They can fatigue, but they don't fatigue easily. You can actually see if this is tetanus. You can see that with slow oxidative fibers, they hardly ever fatigue. The examples are the muscles that are responsible for your posture, right? It's good that they don't ever fatigue. It helps you stand up properly on a regular basis. Slow oxidative skeletal muscle. Now, fast glycolytic, let's just go to the 2 extremes. These are very powerful muscles, extremely powerful. They have a large diameter, lots of myofibrils, and they're pretty much used for quick bursts, jumping, or short sprints. But you can see here they fatigue quite easily. All right, and then the intermediate ones, you can see they fatigue, but over a longer period of time compared to the fast glycolytic, but not, right, the slow oxidative hardly ever fatigue. Now, every single skeletal muscle, whole muscle within your body, actually has all 3 types. All right, since we're running out of time, I'll come back to this. I just want to show you, I really like this diagram. It really shows you some of the major differences between the slow oxidative and fast glycolytic. And it explains why there's differences in fatigue. So let me show you right here. These red fibers, this is a cross section of a whole muscle. And here's the connective tissue. The red muscle fibers are the slow oxidative. They have lots of myoglobin. That's a molecule that can hold oxygen. And it actually gives it a red color. Right? This would be any kind of red meat. That if you were eating like a chicken or something like that, that would be any kind of red meat, is more about slow oxidative fibers. So it has a lot of myoglobin. It has a lot of mitochondria. And you can see here it's a rich blood supply. These black dots are actually capillaries, blood supply. So you're getting a lot of oxygen, and it's generating a lot of ATP. Small diameter, though. The large orange fibers are fast glycolytic. You can see that they don't have a lot of myoglobin, not a lot of mitochondria. There's not much of a rich blood supply, but they are very large. They have lots of myofibrils, so they're very powerful. So this is a nice diagram kind of detailing the differences. If you want to take a look at the real difference between slow oxidative and fast glycolytic, this kind of details all of it. And this is where we'll begin on Monday. Have a wonderful weekend, you all. Oh, wait. Before you go, I wanted to show you one more slide here. Just in case, I want to show you guys, if you actually get in a situation where you have to, this is the secret to crocodile wrestling, just so you know, if you ever get in this situation, crocodiles are mainly made up of fast glycolytic, right? They have very powerful muscles, but they fatigue easily, so if you can just hang on for 5 minutes, you'll wear it out and you'll win, okay? All right, 5 minutes.
I just got back from San and I had my yoga interview. I think it went well. So I should hear back by next Tuesday from a couple of so I'm excited. Thank you. But as far as this class goes, we had 2 problem sets.
Yeah. So I know that you've been, traveling.
Yeah.
You could probably get on the campus for this, so let's kind of explore that.
Yeah.
But if you're having some issues putting it all together, you want to just come in, please make noise. If you want to come in and just talk about it and maybe do some
Yeah.
and then just come on into my office. I'll sort it all out and I'll give you those things and obviously we'll have this. But if you can do that online this weekend on your phone,
Yeah.
I can maybe, you can shoot me a message and you can assign those top hat questions for me for home.
Okay.
And I can actually do that through top hat.
Okay.
You'll see it and then you can just answer.
Okay. That'd be fantastic. Thank you.
Yeah, congratulations. Hi, I got your email. Okay, perfect. Okay, so yeah, I have to figure out, we have the labs on Tuesdays or Thursdays, but it looks like that you can do anything after 10 or 2 o'clock on either day.
Yes.
Perfect. Okay, so we have 2 sections usually in that. We have a 3 o'clock and a 5 to 10.
Okay.
So which one do you think you would probably prefer?
Probably the 3 o'clock.
Okay, then I'll go ahead and put you down for that.
Okay.
Okay,
Thanks. And I also have a question.
Yes. Oh, yeah.
So the difference between the intensity relationships for the length, not the length, for the frequency versus the first intensity.
Yes.
So is the only difference the amount of frequency to this?
Right. So when you're looking at the intensity force relationship, it's only a single twitch.
Okay.
You're allowing it to contract and then you're allowing it to relax all the way back down to a base.
Oh, I see.
Just a single twitch.
Okay.
Frequency force is actually increasing the frequency, so you're not allowing it to relax completely, and then you're going to get subnation.
Okay. That makes sense.
Yes.
Okay.
Yes. Yeah, you're welcome.
Hey, like 2 weeks ago, I missed class because I had the career fair and I got on the wrong bus. And I emailed you about
Okay.
the cystic fibrosis
Okay.
problem set. I took scans
Okay.
of it.
Yes, I have to go through my emails and put all that in, and I do have a few other assignments that people have given me.
Okay.
Magna's great about putting this. I'm not so great, so I'll take some time this weekend to update.
I still have it in paper form too.
If you
want
send
to
it
for
to
your
me,
size.
yeah, no, I think if you just give me a chance to go through the emails and update the grade book, that's great. If you actually want to send it to me again and at the top of my email list, then I don't have to search for it. That's fine too.
Yeah, I can do that.
But I'm gonna try to get all of that in this.
All right, sounds good.
Yeah, thank you. Thanks for the reminder. You can, and a lot of times those muscles are different than those micro tears. This is actually more micro tears are very pretty much in the water. But if you get a full muscle, a lot of times that has to be and it may be more of a tear than a micro tear. Yeah, that can be problematic. So you're right. You can lengthen the muscle to actually cause some injury, whether it's at the muscle, and that would be kind of more associated with that muscle, which then would be some time to heal.
So the muscle
Yeah. So when you're talking talking about preload, it's less about skeletal muscle because your muscles are already off in the length the way it's attached, and you don't detach the muscle and change the preload. But for the heart, it is different. The heart is when you stretch the muscle because you force more blood into it, you're actually stretching it towards the heart. And you hardly ever stretch it beyond the flow, unless there's a situation where there's an error. Like heart failure changes the dynamics of preload, and then that can be a problem. But you wouldn't ever pull your heart muscle. Yeah. So that's been, and we'll talk more about that too when we get to the cardiac section. Revisit preload and talk about the starting mechanism, which is really all about preload and the length of concentration. If you want to skip ahead and kind of those steps. But that's where you really go into varying amounts of blood into the heart and the changes in preload. Yeah, good question. Thank you. Yeah. Oh my gosh. No, I didn't even notice.
Okay.
I didn't even notice.
Alright, okay.
Yeah.
And also, just wanted to ask, it's like how does a muscle like, you talked
Yeah.
about the preload, but like,
Right. So here's how to think about it. With preload, you're actually talking about the tension ratio. So you're varying the muscle, and then you're varying the tension. With afterload and those little weights that were in the muscle, that's what the muscle has to work against when it's after stimulation. When you stimulate it and contract it, that those weights are what the muscle is working against.
so is that the light load or?
Yeah, so they're varying afterwards. You have a light load and then you have a heavy. Usually the velocity is going to be different.
So it's like, yeah,
Oh,
just.
let me, sorry. Do you guys all want to move in the back? Because I know Suzanne's going to be here.